So today we are going to talk about Taxol. This is the syllabus outline, and we are going to talk more about it later. This is the structural formula of a Taxol, and later we are going to analyze it. So Taxol is under the big category of taxols. It is a member of a class of anti-cancer drugs derived from U3 products. So from these products, you can also get another form, like another category of called Taxol. Yeah, next one. So the discovery of Taxol starts from early 1955 when U.S. National Cancer Institute set up a national service center to look for plants that can cure cancer. So later in 1962, they found out that the bark from a single Pacific yew tree may be helpful to treat cancer. So in 1964, they found that these samples contain some elements that is useful and cytotoxic. Later, they identified the active ingredient in these bark samples and named it as Taxol in 1967. Yes. Next one. So, Taxol is a very chiral drug. It means that the carbon is attached to 4 different R groups, and in the structure of Taxol there are 11 chiral carbon centers. So there are a lot of enantiomers, and it was later approved for use as a chemotherapeutic agent in 1992. So in the US, the FDA has approved it as treatment for breast, pancreatic, and non-small cell lung cancer. So in UK, it was also approved for treatment for breast, ovarian, prostate, and different kinds of cancer. Next one.
So for chiral drugs, we need to understand enantiomers. Enantiomers are pairs of chiral molecules that are mirror image of each other. Like we learned in organic chemistry earlier, right? It's like your left hand and right hand, they're mirror images. And chirals are defined as a carbon atom that is bonded to 4 different R groups. We will see later on in the structure. And these different enantiomers usually have similar identical chemical properties, but they react differently in a chiral environment, which is how they react differently when you put it into your body. Remy will explain more about how the effect that this has on you later. Secondly, chiral environments are essentially an environment in which 2 enantiomers can be distinguished due to the way that the isomers react with the environment. So for example, enzymes and receptors in your body, when you insert Taxol into your intravenous vein system, will lead to different enantiomers having different physiological properties. Next. So to explain this, the way that enzymes are affected, you can see that only one enantiomer is biologically active. So you can see that on the left side, the enantiomer is active because it binds with the A, B, and C side correctly. But if you rotate it to get this mirror image of enantiomers, it is inactive because the C and the A side are no longer corresponding to the appropriate active sites. As such, they are no longer active and they no longer give the desired the physiological effects. Next. The way that we can differentiate enantiomers is using a machine called a polarimeter. So it's a device for measuring the angle of rotation of plane polarized light caused by optically active molecules. So different enantiomers, by virtue of having different orientation, react to light in different ways. By different ways, when you shine a light through a polarizer, the plane polarizing light will then bounce off the sample, which will tilt or polarize the light in a certain degree, and by measuring this degree and observing how much this degree, we can determine what enantiomer this is. So next, chiral drugs produced by biological synthesis reactions that produce one enantiomic form, right? Because when we want to obtain an enantiomic drug, we will try to harvest a drug from a natural source and then try to extract a single isomer from it. However, most often the times when we produce synthetically or artificially certain drugs, we tend to get a mixture of enantiomers. In this mixture of enantiomers, if it is in equal ratio, it will be called a race mate, right? Equimodal mixture of both enantiomers. However, given that enantiomers react in the body in different ways, it's important for pharmaceutical companies to isolate and extract only one type of enantiomers.
Yes, sir. So there is increased importance into the research about stereochemistry after the thalidomide tragedy because a lot of deformed babies were born, and out of the two isomers of thalidomide, only one of them is helpful. Yes, the other one is toxic and cause undesired effect.
So what we do now is to isolate the desired enantiomer from its race mate mixture. The structure of the Taxol has 11 chiral carbon centers. We need to use this process called a chiral auxiliary. So a chiral auxiliary is an active substance that is put into an organic synthesis so that it can be carried out asymmetrically with the selective formation of a single enantiomer. You block everything except one specific reaction pathway so that you can only have this reaction going around, developing in a specific way such that the exact structure that you want is formed. And this process is described below in 5 steps. 1. The chiral molecule binds to the reactant. 2. One reaction site is physically blocked through steric hindrance. 3. This creates the correct stereochemical conditions required to force the reaction to produce as desired, in other words, of the desired enantiomer. 4. The auxiliary is then removed once the specific enantiomer is set, and afterwards the auxiliary can be recycled.
Okay, so another way that Taxol can be developed is from nature, and this was like the way that it was originally developed, because from 1967 to 1993, almost all of the Taxol was derived from the bark of the yew tree, but this killed a lot of trees in the process, obviously, because once they cut the bark, the tree would die. And the problem was that the Taxol tree, the Pacific yew tree, took very, very long to grow. So obviously this wasn't like the most sustainable. So there was a very high demand for Pacific yew tree bark, but a very low and slow generating supply of it. Yes. So you can see here the benefits of nature versus synthesis. So if you produce Taxol from nature, there's a very high cost of extraction but a very low yield, and 1 gram of Taxol requires 100-year-old yew trees, and only a single enantiomer is produced when you harvest it from the tree naturally. So that's good, but you need so many trees. And the Pacific yew trees also provided a habitat for a lot of animals in nature. Whereas the commercial synthesis of Taxol is actually there are not many things good about the commercial synthesis of Taxol, because it's a very complicated process, because you need like 30 steps to like derive one molecule, and there's still a very low yield, and also it produces toxic waste, and you get multiple enantiomers. So the thing called semi-synthetic synthesis came about. So basically, when you have a lot of steps to yield a product, you get a very low yield. So what the scientists were trying to do is like to try and reduce the number of steps to get a higher yield of products. So what they figured is that they would get the related compound from the leaves of the yew trees, which grows back faster and can also be harvested more easily. Yeah, so this became known as the semi-synthetic synthesis, which began with a precursor from nature and ended up as Taxol. So the process basically required the conversion of 10-deb, which is the related compound from the yew trees. Yeah, and that was converted to Taxol using 13 solvents and a range of other organic reagents. Okay, so we have a video on it. You can watch it yourself. Yes. Okay, now we can look at the pros and cons of semi-synthetic synthesis. So overall, there's a higher yield, but it does involve a lot more toxic solvents and chemicals. And while it is sustainable to harvest the leaves, allowing them to regenerate more quickly than if you were to harvest the bark, it still does produce a lot of toxic waste. Yeah. Okay. So now we will look at how Taxol actually cures cancer. And to understand how Taxol cures cancer, we need to know what cancer is. So cancer is basically a disease caused by an uncontrolled division of abnormal cells in a part of the body. So when cancer develops, the cells do not die when they're old or damaged, but they divide instead. So new cells aren't formed, but the harmful and dying cells are replicated. So this forms tumors, which cause a lot of damage and are harmful to the body, causing cancer. And you can see the diagram here. Yeah, okay. So Taxol is used as a chemotherapeutic drug in chemotherapy, which is the treatment or control of disease by chemical agents, and this is obviously usually used in the context of cancer treatment. So Taxol is a cytoskeletal drug that binds to beta tubulin. You just need to know the tubulin part, which is a protein in the main, and is the main component of microtubules, which form spindles during cell division. So what Taxol does is that it stabilizes the microtubules involved in cell division and prevents them from disassembling. So the chromosomes can't be moved around the cell. And this usually happens around metaphase of cell division. Yes, as you can see here. Because the spindle fibers can't be disassembled, mitosis stops at metaphase and the cell can't divide. So I will draw a diagram for you to see. Okay. Okay, this is called anaphase. You don't need to know that, but yeah. But because Taxol is a cytoskeletal drug, it will bind here, right? Yeah, and prevent this from breaking. So it's stuck because of Taxol. Yes, and this thing is made of tubulin, which is what Taxol binds to. Yes. So this blocks the progress of mitosis, and the prolonged activation of this mitotic checkpoint triggers apoptosis, which is normal cell death, or it causes the cell to revert back into G phase, which is somewhere here, yeah. So basically this cell is stuck here and it doesn't divide, and it either goes back to being a cell that isn't undergoing cell division, or it just dies, which is what we want, because we want to stop the spread of the tumor. Yeah. Okay. So we have some alternatives for producing Taxol, and basically is they found some fungi that can produce Taxol in fermentation reactions, and we also have plant cell fermentation technology. So when the Taxol is extracted directly from a plant cell culture and purified via chromatography, that's the process. And these processes are good because they eliminate the use of many hazardous solvents, which is green chemistry. Yes. And basically it's very important for us to develop more green ways to develop Taxol, because the demand for chemotherapeutic agents is always increasing, because more people are getting cancer because of lifestyle problems and stuff. So there's a very big pressure on the industry to produce and find a sustainable source of chemotherapeutic agents.